We are here again now, guys. So there is a little to know from here, because we understand mitosis. So this meiosis 2 now is also just divided into those four stages. So we are still going to have, in this case now, we'll call it Prophase II with regard to meiosis 2. Then Prophase II, we are going to have Metaphase II. Metaphase II, then here we are going to have Anaphase II. Then finally we are going to have Telophase II. So at the end of Telophase II, we are going to have we're going to have those four cells which are not identical, like this. Okay. So we start again in Prophase 1. What happens in Prophase 1? The same thing that happens in Prophase 1 of mitosis. It's the same that happens here. But at this point now, think of two cells undergoing the same cell division at the same time. So we have these two cells. Then this one now is having those its own chromosomes. These chromosomes, as we know, that they are scattered everywhere in the cell. Then we saw the nuclear membrane surrounding these chromosomes at the end of telophase 1, like that. Then it is these now which have entered meiosis 2. So when they enter meiosis 2, they start with the prophase. So in prophase, the same will happen. The nuclear membrane will disappear, then the chromatin will decondense by coiling so that it can become thick and short. That is the end of prophase 2. Then the cell will enter metaphase. So in the metaphase, we get to see now these chromosomes aligning themselves at the equatorial plate. Will align themselves at the equatorial plate. This is happening in simultaneously in these two cells which we have here. Then those will come here. We have them. The sister chromatides are still attached at the centromere. But don't forget that these chromosomes at this point are recombinant. I was supposed to be showing those recombinant chromatides, but it's not possible for me. What we mean by recombinant is that there is still that portion which came from the paternal chromosome. This one, there is still that portion which came from the maternal chromosome. That's what we mean. That's how these chromosomes are existing at this point. Then, of course, we have the centrioles to the opposite pole here. Then they are sending their spindle fibers to the kinetical of these chromosomes. The same is true with this side, or the same this side. Here now we are in a metaphase 2 of meiosis 2. Then when this is completed, the cell then will enter the anaphase 2. So in anaphase 2, we know what happens in mitosis. In the anaphase of mitosis, the sister chromatides are going to separate, thereby letting each one of these become an independent chromosome. So the same is true. That's why here I'm showing two cells. Then now what we have is that the sister chromatides in here have separated. So we have that one there here. Then this one was a sister chromatide to that chromosome, like that. The same is true here. The sister chromatides now have separated to become independent chromosome. And those independent chromosomes will move to the opposite poles. Just know that that is the anaphase. Of course, here we still have these spindle fibers attached to the kinetical of those now which have become independent chromosome. The same is true here. What is happening here now, guys? This is the anaphase 2. the Anaphase II. When this is achieved, one has a question? Go ahead. Okay, so it's very unfortunate that we don't have brother chromatide. No one understands why they chose to call these as sister chromatides, or to call this cell which you start with as the mother cell. There's no father cell, there's only mother cell and the sister chromatides. That is how unfair this world can be. Okay, guys. Yes? Okay. So you're trying to defend yourselves. That sounds cool. Okay. So, guys, the differences here between Anaphase 1 and Anaphase 2. Anaphase 1, or maybe before I give you this information, who can try to give us the difference? Though I've already mentioned this. I remember mentioning it earlier on. What is the difference between Anaphase 1 and Anaphase 2? Guys, if you can't answer this question, then I have to conclude that you guys are not following what is happening here. Yes? Yes? Yeah. Yes? Yeah, exactly. So that is the difference. Remember, you guys, I said that when these homologous chromosomes are entering the anaphase 1, they are still attached at their chiasma point. We still had this. These homologous chromosomes were still attached at the chiasma point. So maybe I show something like that. Okay. So what separated in anaphase 1 where this was a pair of chromosomes which we call the homologous chromosome. Then now what separated it was these homologous chromosomes which separated from each other. The point which separated was the chiasma point. So they detached from each other, thereby moving to the opposite side of the cell. So we had this. So the major difference between anaphase 1 and anaphase 2 is that in anaphase 1, it is the homologous chromosomes which separate, whereas in anaphase 2, it is the sister chromatides at the centromere where they separate. So now what we are looking at this point is that this chromosome is separating there at the centromere. This now comes to make up anaphase 2. This is the major difference which is there. And this is why we said that meiosis 2 is very similar to mitosis. Because in mitosis we only see the sister chromatides separating from each other at the centromere. But in anaphase 1, we don't see the centromere separating. We only see the the homologous chromosomes separating from each other, which is not there in mitosis. So those are the major points to understand. So where we have reached now, it's at the anaphase 2. So here we get to see those sister chromatides have separated and moved to the opposite poles. And you and me, we agreed last time that when the sister chromatides separate, these becomes independent chromosomes. So that's what we have there. These are independent chromosomes. Then now what next? Telophase. Telophase 2. So telophase 2, this will be followed by cytokinesis, division of the cytoplasm. So there is a decondensation which takes place here. The chromosomes are going to decondense so that they can become thin and long. Then also the nuclear membrane will reappear here. So what we are going to end up, guys, is this. I'll only show, because these are going to divide like that, thereby giving us two cells. So what we have now here, we are going to have, this one is going to produce these two cells. The same will be true with this one here. It's also going to produce two cells. But in terms of the number of chromosomes, this here is only going to have those two chromosomes, like this. This one also like that. The same is true with this one here. But remember, guys, that these chromosomes are the recombinant chromosomes. They are containing both genes from the mother and the father. Then also see that the nuclear membrane appears here. So these chromosomes are surrounded by the nuclear membrane, like that. The same is true. Here also we get to see two cells which have been produced there. The same, these chromosomes are recombinant chromosomes. So I was supposed to be showing half here containing the portion from the mother, like from the father, like that. So that is what happens now. So what we have now here, guys, is this. Okay, so there we also have the nuclear membrane. Here we also have the nuclear membrane. So what we have now here is this one has got diploid number of chromosomes. Diploid number of chromosomes, we agreed that it's denoted as N. The cell, the mother cell which we started with, the zygote, had 2N, the diploid number of chromosomes. But this now has been reduced to half, just N. Then we also get to see that there are 4 cells here which are produced at the end of meiosis II. These cells produced here, they are not identical to each other as we used to call in mitosis. These are not identical. Who can guess the reason why these cells are not identical at this point? We are saying that these cells which have been produced here, these 4 cells that we have, they are not identical from each other. So I was saying that who can guess the reason why these cells are said not to be identical? These two which we have here, they all come from the same cell. This one here on top. But we are saying that they are not identical to each other, despite them coming from that this single cell. The same is true with these are not identical to each other. Then overall, all of them 4 produced, none of it is identical to the other. And none of these also is identical to their mother cell. So what is the reason why we call these cells produced here not to be identical to each other? What could be the reason? Yes? Yeah. No any other reason apart from that. So these cells are said not to be identical because remember in Prophase I, there was crossing over. So there was a gene shuffle. Gene shuffle. So the gene which are in this chromosome cannot be the same as the genes which are in this chromosome because we had those 23 pairs. Each one of those 23 pairs had close-matched some of their portions with the chromosome from the other pair. So that close-matching is what makes these cells produced here not to be identical to each other. So the reason is because of cross-matching which took place in Prophase I. That's why they are not identical. But they have got haploid number of chromosomes. that is half. So this now what we have produced here, if it is in males, we have produced what we call sperms. If it is in females, we have produced what we call eggs. So for you who are doing medical related fields, next year you are going to start from here again, discussing what happens down. But your first year, here we end. This was the goal. Then this is the end of meiosis II. Then what do you think will happen if the homologous chromosome did not separate, or the sister chromatids did not separate? Let's say here, this chromosome, instead of separating its sister chromatids, then the whole of it was just put to one side. What do you think will happen? Exactly. So these guys, if it is in human cells, we are saying that these have got 23 chromosomes. This is standard. This one, this side also have got 23 chromosomes. But think of, let's say in meiosis 1 there was those did not separate. Okay. So when the homologous chromosome fails to separate, we call that condition as a non-disjunction. These are your stuffs. So there is what we call non-disjunction. So non-disjunction, this is when the homologous chromosomes or sister chromatids fails to separate during anaphase. That is what we call non-disjunction. So non-disjunction can take place in meiosis 1 or in meiosis 2. But these have got different effects on the cells produced. When there's a non-disjunction, guys, the cells produced are not going to have those standard number of chromosomes, which we are saying 23. They are going to have a plus or a minus. That is the effect of these non-disjunctions. So there are different diseases, genetic diseases, that are caused because of non-disjunction. But those diseases, most of them which are caused because of non-disjunction, they are not genetically inherited. So why they are not genetically inherited? Because it's just an error of homologous chromosome failing to separate during an anaphase, or during a sister chromatid failing to separate during anaphase 2. So what I was saying is that there are different types of effects that can result because of non-disjunction. So let's start with non-disjunction in meiosis 1. What effect is going to be there? So think of, we'll start from anaphase, because that's where this non-disjunction can take place. So this here is going to be anaphase 1. So we said that at anaphase 1, the homologous chromosomes separate. So we have this. I'll show these two chromosomes. So we have this chromosome here. Okay. So that is the homologous pair of a chromosome. Another homologous pair of chromosome is here. Okay. These are attached at the chiasma point. Then of course here we have those. Okay. So we know that at the end of anaphase, what do we expect to see? What we expect to see at the end of anaphase, it's a situation where we only have those chromosomes on this side, and these chromosomes also this side. This is in a normal, okay, in normal situation, this is what we expect at the end of anaphase. The homologous chromosomes have to separate completely. But in a case of non-disjunction, what will happen is this. Instead of this cell looking like this, it will look like it will look like this. So we are going to have a single, let's say this pair separated completely like this. Then we also have this pair of chromosomes on this side. Then now we have this another pair which failed to separate. Then hence it moved to one side of the cell. Like this now. This is what we call non-disjunction. So we get to see that these did not separate. That pair of homologous chromosomes did not separate. Where are these down here separated? So what will happen now here? At the end of telophase, there's going to be this. These two cells are going to be produced. So these two cells are coming from the same cell. Quite alright, they have to have haploid number of cells like that. But because there was non-disjunction, what we are going to see is going to be this. Okay, so these guys, here we are in the Anaphase II. Then we have passed telophase. So in telophase, we have these two cells, like that. These two cells will also enter meiosis II. So in meiosis II now, there's going to be separation of what? There's going to be separation of nucleotides. Oh, sorry, chromatides. The separation of chromatides. So what will happen is that this cell, the chromatides are going to separate. They are going to separate thereby giving us two cells. Then this side also are going to give us two cells. But now what will happen is that these cells here are only going to have a single chromosome. A single chromosome because that one there is the one which is going to separate like that. Then this side, there's going to have an extra number of chromosomes. So these are going to have 3 each. Why they are going to have 3 each? Because we are going to see these sister chromatides separating in Anaphase II, that one also separating, this one also separating. So these are going to be 3 chromosomes in here, then another 3 chromosomes in there. These guys are the conditions which come about because of the non-disjunction, failure of either homologous chromosome taking place or sister chromatides failing to separate. So what we see here now is that this one is n minus 1. It's less than a chromosome there. That's why it's n minus 1. So it is 23 minus 1. So meaning that this cell now has got 22 chromosomes. The same is true with this one, n minus 1. Where are these two here? They have got n plus 1. This one also has got n plus 1. So we can see that here there is no normal cell which has been produced. Now these conditions where a cell ends up with an abnormal number of chromosomes are what we call an euploid. You come across this again in genetics. An euploid, this is an abnormal number of chromosomes in a cell. So all of these conditions which we have here, which come as a result of non-disjunction, are what are known as an euploid. So there are 2 types of an euploid. There's too much data here, guys, to understand. So an euploid, we are saying there are 2 types. There's what we call monosome and the trisome. Monosome and trisome. So this monosome, it has got less number of chromosomes, n minus 1. That's why we call it a monosome. Whereas a trisome has got an extra chromosome, it's n plus 1. So in this case, these are trisome, these are monosome. So there are genetic diseases which come as a result of these aneuploid. The common example is Down's syndrome or Down syndrome. So Down syndrome comes as a result of non-disjunction which takes place at a chromosome 21. There's non-disjunction at chromosome 21, thereby the cell has got less number of chromosomes. So it only has got 22 pairs in that case. So that is a monosome. Then there are also some diseases. We are going to discuss genetic diseases toward the end of Mendelian genetics or post-Mendelian genetics. There are diseases such as Turner syndrome or Klinefelter, which has got an extra chromosome. So those have got some people who have got 47 chromosomes instead of 46. That comes as a result of these non-disjunction which takes place, failure of either these sister chromatids or sister chromatids separate or the homologous chromosome. So that is the effect of these non-disjunction that can which can occur. So this is when there's non-disjunction in meiosis 1. When there's non-disjunction in meiosis 2 now, it is the failure of these homologous chromosomes or these sister chromatids to separate. So meaning that in that case now, Anaphase II is going to be completed just normal. So it will be completed just normal. Then instead now of in Anaphase II, these sister chromatids to separate at the centromere, one chromosome is not going to separate. So what we'll have is non-disjunction. When non-disjunction takes place in Anaphase II, we have two cells which are going to have normal number of chromosomes. So two are going to have normal number of chromosomes, whereas two are going to have less number of, or one is going to have, this one is going to have 1, then the other one is going to have 3. This is the outcome. So here we have this one n. N we know is the haploid number of chromosomes. This is n minus 1. This is n plus n plus 1. N plus 1. So this is when non-disjunction takes place in meiosis 2. So you can see that when non-disjunction takes place in meiosis 2, there is a probability of producing 2 normal cells and 2 abnormal cells. Whereas when non-disjunction takes place in meiosis 1, all the cells are going to be abnormal, as we have seen here. So that is a very simple task to complete. What you can do, if you want to come up with how we get to have 2 normal and 2 abnormal, just think of a cell which completed meiosis 1 very normal, then these homologous chromosomes failed to separate. Sorry, the sister chromatids at this point failed to separate. So we are going to have this cell which is going to have this chromosome which separated, then you have this one which did not separate. Then this side, we only have that single chromosome. This is what is now going to give us this scenario which we have here. Where? This scenario which we have here. Any questions, guys?